Hello there. Welcome to Fundamentals of Biology, Lesson 6, where we're going to be looking at proteins. So this is the fourth of our four classes of biological molecules. So we've already looked at carbohydrates and then lipids and then also nucleic acids. So today we're going to finish that section off by looking at proteins. And if you remember what I said in the previous session where we looked at nucleic acids, I mentioned that the nucleic acids and proteins are intrinsically linked. So nucleic acids, so DNA and RNA, are involved in the production of proteins. So DNA is the blueprint, the plan, the instructions, if you like, for making proteins. And then RNA actually interprets those instructions and produces the proteins. So today we're gonna start talking about proteins, what they are, what they do, and then we'll also have a look at their structure as well. So the objectives for this session, then I'm going to start by doing a quick recap of functional groups that I've done in previous sessions. Then we're going to go on to look at amino acids, which are the monomers that are joined together to make our proteins. And amino acids are joined together by peptide bonds, so we'll look at those. And then we'll finish off by talking a bit about protein structures, because proteins are really large, complex, three-dimensional molecules, and their structure is really, really important. It's what enables them to do their jobs. So this is the same slide that I've shown you previously, and it just highlights four of our seven functional groups that we encounter in biochemistry. And if you remember, down here we have the hydroxyl group, the oxygen and the hydrogen, which we've encountered quite a lot already. So if you remember, when we see our hydroxyl groups on parts of molecules, this is often where we'll see dehydration synthesis occur. We also up here have our carbonyl group, so a carbon double bonded to an oxygen. And then over here we have a carboxyl group, which is our carbonyl group attached to a hydroxyl group. So if you remember, we saw these in the session that we did on lipids, and we actually saw these at the ends of our fatty acid chains. And it is through the hydroxyl group, which is part of the carboxyl groups, that dehydration synthesis occurred and enabled our fatty acids to attach to our glycerol molecule. So we're going to see carboxyl groups again today. If you remember, the full name for the carboxyl group is the carboxylic acid group. And then down here we have the amine group, or sometimes referred to as the amino group. And that's a nitrogen covalently bonded to two hydrogens. So I've mentioned just now that one of the things that we're going to look at today is the monomers of proteins, and the monomers of proteins are amino acids. And here we can see two functional groups, an amino group and a carboxylic acid group. So an amino acid. So these two functional groups are going to feature prominently as we go through and look at and talk about amino acids. What you can see here is a three-dimensional rendering of a protein. And what you can see here is that it's a really, really complicated structure. They tend to be big molecules, proteins, and they are a very, very specific, or they have a very specific three-dimensional shape. And it is this shape or structure that determines their function. And this is something that we see a lot in biology, this process where structure equals function. So the proteins, to be able to do their job properly, have to have a very, very specific structure or shape. And as we go through this, I want you to keep that in mind, and I will keep coming back to it and talking about it as we go through, because it is the underlying key to what proteins are and how they do what they do. It's all down to their structure. And if you remember, the instructions for building our proteins is what's written in our DNA. So DNA is what ultimately determines the structure that our protein will have, and therefore the job that protein can do. What are proteins? What do they do in life? Well, as you can see on this table, they have a huge array of functions within living organisms. So this is not an exhaustive list. I've just chosen some of my favorites as examples. Essentially, if you were to take a human and you were to dry us out or reduce us to just dry matter, over 50% of what we are made up of are actually proteins. And proteins are at the heart of pretty much all of the biological functions that we do, from muscle contractions to nervous systems working to digestion of our food, the physical structures that make up our bodies. They are all heavily dependent on proteins. So let's just quickly go through this, well, not necessarily quickly, but we'll go through this table and I'll give you some examples. So at the top here, we've got enzymatic proteins, so enzymes. And as we can see here, their function, selective acceleration of chemical reactions. So enzymes are so important that I'm going to do a whole session on them in the next lesson. And essentially, enzymes are needed because our life is completely dependent on chemical reactions, particularly when we talk about things like digestion and our metabolisms, things like that. Even the conversion of food into energy that the body can use. It's all completely dependent on chemical reactions. And at normal body temperatures, those chemical reactions would just happen far too slowly to support life. So enzymes are important or necessary for life to function. So what I've given you here is just a few examples that we find in our in human digestive system. So we've got sucrase. Well, I've mentioned this one before in our session that we did on carbohydrates. So sucrase is an enzyme that will hydrolyze or break down sucrose, normal table sugar. And if you remember, sucrose is a disaccharide. It will break, so sucrase is an enzyme that will break sucrose down into glucose and fructose. The next one here we can see is amylase. Well, amylases are enzymes that will break apart starch. So if you remember, starch is a storage polysaccharide. So it's lots of glucoses all joined together. Well, amylase is an enzyme that will break down that starch into the single glucoses, and then we can use them to produce energy. Lactase, also one that we've mentioned, used to break down lactose that we get from milk, and lactose can then be broken down into glucose and galactose. And then we have pepsin, which is a stomach enzyme that begins the breakdown of proteins in our food. There's just an example. We have thousands of enzymes in our bodies that carry out specific chemical reactions, and we'll talk about some more examples of those in the next session. We also find proteins making physical structures within our bodies and throughout other parts of the natural world. And structural proteins offer, as you'd imagine, physical structure and support. So examples, we've got collagen and elastin. These are both really important proteins in our skin as examples. So collagen gives our skin strength and flexibility, and elastin actually gives it skin the elastic properties, so it can snap back into its original shape. And elastin is one of those proteins that we actually stop producing as we get older. And that is one of the reasons why we start to get wrinkled as we start getting older, because the elastin isn't being produced and sort of repaired and replaced as we get older, and it's broken down by oxidation and things like UV damage from sun exposure, things like smoking and other environmental pollutants. All of these things over time will break down the elastin, and as we get older, that elastin is no longer replaced. So our skin starts to appear more wrinkled, and unfortunately, that's just something that happens to us as we get older. I've got a little bit more bad news. That replacement of elastin starts to take place around sort of the early 20s. So around the early 20s, that replacement starts to slow down. And then this is why by the time you get into your 30s, you can start to see wrinkles appear. And depending on life choices and things like that, that will occur more rapidly in other people. So if you are a sun worshipper and you spend a lot of time sunbathing or using sunbeds, and if you smoke, if you sort of don't stay hydrated, and all of those kinds of things will increase or speed up the degradation of your elastin. So it will lead to rapid aging, or the appearance of rapid aging in your skin anyway. That was a bit depressing, wasn't it? Sorry about that. Anyway, another structural protein is spider silk. Spider silk is not something that we often think about, but it's actually just made up of protein, and gives it incredible strength and sort of resistance to breaking. The next group we've got here are storage proteins, and storage proteins are really, really important for developing young, particularly developing embryos. Basically provide a storage of amino acids and proteins that developing young can use to build their own proteins as they're growing and developing. So two good examples are ovalbumin, which we commonly know as egg white. So egg white or ovalbumin it's a store of amino acids and protein that the developing young can use to actually build their own proteins. In mammals, we actually have something similar, and it's called casein. And casein is found in milk. When female mammals produce milk, that milk provides, or it contains casein, so the developing young can use the casein to actually build their own proteins. Next one down, we have transport proteins. So as you'd imagine, they transport other substances around the body and in and out of cells. Good example here is hemoglobin. So hemoglobin used to transport oxygen and CO2 around the body. But we also have, on a much smaller scale, we have specialized proteins embedded in the membranes of our cells. And we'll look at these in more detail in a later lesson. But a classic example in biology is here. It's called the sodium-potassium pump. And this is incredibly important in maintaining the function of our nervous systems. And like I said, we'll look at that in more detail in a later lesson. Okay, next group, hormonal proteins. We talked about some other hormones before when we talked about lipids. So we had the steroid hormones, so estrogen and testosterone, cholesterol, progesterone, and vitamin D. Well, those are examples of lipid hormones or steroid hormones. But we also get a lot of hormones that are proteins. And these coordinate an organism's activities, long-term, long-acting activities, things like metabolism and growth and the regulating of blood calcium levels, blood glucose levels, things like that. Good examples are insulin and glucagon. These two work against each other. They are antagonistic hormones that help to regulate our blood glucose levels. And then we have oxytocin here, which is often referred to as the cuddling hormone. It's sort of released to increase bonding between individuals. So it's something that you can experience at the start of a new relationship, or it is produced in parents when they've just had a child, to increase the bonding between those parents and the child. There has been some studies that have shown that in humans anyway, when a couple are about to have a child, there's a drop in testosterone levels in the male and an increase in oxytocin levels. So if we think about that, the high levels of testosterone that we see in some men, or a lot of men, is the testosterone is there one of the things it does is it increases aggression. You don't necessarily want that when you've got a new baby coming about. So testosterone levels are shown to drop, and oxytocin levels, so oxytocin, which increases bonding, those levels increase to increase the chances of the male bonding with the child, which from an evolutionary point makes real sense, doesn't it? If the child is going to survive, it makes sense if there are two parents looking after it. So that's a really good example of hormones changing in concentrations at different times. Next group down, we have receptor proteins. And receptor proteins are sort of all throughout our body and embedded into our cell membranes, things like that. And they enable our nervous systems to respond to chemical stimuli. So these will be typically poking out of our cells, embedded within our cell membranes, and they will basically be there to for scent molecules and other molecules to attach to that enable our nervous systems to detect and then ultimately respond to stimuli. We also have another group here, so contractile or motor proteins, and these enable movement. So when we look at our muscles, muscle contractions are carried out by two specific types of protein, and they're called actin and myosin. And these overlap each other to enable muscle contractions in something called the sliding filament model. And this is something, again, that we'll look at in a later lesson when we look at tissues and we actually look at the musculature system. So actin and myosin are both types of protein that enable our muscles to contract. We also then have defensive proteins, so produced by the immune system, and our immune system helps to protect us from disease and against pathogens. So a good example here are things like antibodies. And antibodies are moving around in our bodies, and they have proteins that have just the right shape and size to attach to specific antigens, which are sort of little markers on pathogens. And then once they're attached to them, they signal to the immune system, particularly to white blood cells, that these pathogens need to be destroyed. So they're really important components of our immune system. So like I said, this is not an exhaustive list. We have many, many more types of proteins within our bodies. Many thousands of different proteins within our bodies. But these are just some examples that give you an idea of the range and scope of what proteins do to enable life to exist. So now we know a bit about what proteins do, we need to start having a look at their structure, so where they come from. We know from the last session that proteins are made through a combination of DNA and RNA. So DNA carries the instructions, RNA interprets those instructions and actually does the building of the proteins at a site called the ribosome. So ribosomes in our cells are the site of protein synthesis. That's where the instructions within DNA are transformed into, or what we actually call translation. They're translated into proteins. But to do that, we need to join together many monomers to make our ultimately make our protein. And the monomers of proteins are amino acids. So what we can see on this slide is the basic structure of a generic amino acid. And at first glance, if you've not seen them before, it doesn't look that pretty. But we can break it down and it's fairly straightforward. In the center of our amino acid, we have a carbon, and you can see it down here. This is referred to as the alpha carbon. It's right in the middle of the molecule. Above it, we have, remember from our very first lesson together, carbon wants to make 4 bonds. So this carbon is bonded to a hydrogen here. And then here we can see it bonded to one of our functional groups, the carboxyl group or the carboxylic acid group. Over here, it's joined to an amino group or an amine group. So that is a nitrogen covalently bonded to 2 hydrogens. So we've got an amino acid. And this part of our amino acid is really important because this is going to form what we refer to as the backbone of our protein. So when we start joining our amino acids together in a moment, this is where we're going to join our amino acids together. So if you can imagine a long chain of amino acids, say 1,000 amino acids long, they're all going to be joined together through this backbone. But what you can also notice down here is this thing, what we call an R group or a side chain. So when you see an R like this, it just means that there's something else, another molecule is going to be added onto there. So we can refer to it as an R group, or we can refer to it as a side chain. It's one of those sort of annoying things in biology where we have two names for exactly the same thing. So you can call this an R group or a side chain, and another biologist would know exactly what you mean. The backbone of the amino acid structure is always the same. So every amino acid will have this same backbone structure. It's this side chain or R group that is different. It's what is added on here to this alpha carbon that will determine what type of amino acid we have. So how many different R groups are there? Well, I think I mentioned this in the previous session, but we have 20 different naturally occurring amino acids that make up our proteins. So that means we have 20 different R groups. And you can see the 20 different R groups here. Now, you will need to remember all of these. I'm only joking. You don't need to remember them. That would be unnecessarily cruel. But there are some things that you do need to know about them. So we can see here our 20 different R groups that give us our 20 different amino acids. And these are nothing to be scared of. They're just molecules typically made up of carbon, oxygen, hydrogen. Some will have a bit of nitrogen. Some might have a little bit of sulfur. So they're just other molecules. But the important thing that you do need to remember is that they have different properties depending on what molecule they are. So some are hydrophobic, some will be hydrophilic, some will be acidic, some will be basic. So you don't need to worry about sort of aromatic and nucleophilic and things like that. Those are more chemical terms. But the key thing here is that you realize that the R groups that attach to our amino acid backbone, they are different molecules, and depending on what they're made up of, they will have different properties. So some will be hydrophobic, so they will not mix with water. Some will be hydrophilic, so they will happily mix with water. Some will be acidic, some will be basic. What we can see is we're going to get amino acids with very different properties, and that will become important later when we start forming our very, very complex three-dimensional shape of our proteins. So just keep in mind that there are 20 different amino acids, and the backbone of the amino acid, this bit is always going to be the same. So carbon, hydrogen, the carboxyl group, and the amino group, this is the same in all 20 of our amino acids. It's our side chains or our R groups that are different, and there are 20 different side chains, so that gives us 20 different amino acids. Out of those 20 amino acids, 9 of them are what we refer to as essential amino acids. And what that means is this, the reason I mention this is it's something that you might have heard before, this term essential amino acids. And what it actually means is our bodies cannot produce these from other ingredients. So the only thing that we can do is to get them in our diet. So typically this isn't something that we need to worry about as long as we have a reasonable diet in the sense that it is balanced and varied. So you're getting because a lot of these will actually come from vegetable products and sort of leafy vegetables and things like that. But some you can get from eggs and some dairy products, things like that. It's just something to be aware of, that although we can actually typically make a lot of our amino acids from just consuming other ingredients, there are 9 of our amino acids that are essential, and that means we have to get them fully formed in our diet. So if you're interested in nutrition or that kind of thing, it's certainly worth having a look at. It doesn't take much of a search to find what the 9 amino acids are and in what foods we can find them. If you're not worried about nutrition and you're getting a balanced diet, then it's not something that you will ever need to worry about particularly. Now that we know what proteins are and what they do, and now we also know what amino acids are, and the fact that amino acids are the monomers that are going to make up our protein, we need to start joining them together to actually start making a protein. This should be fairly easy now, because this is the same process that we have seen time and time again in the previous sessions. So how did we join glucose and fructose together to make sucrose when we looked at carbohydrates? we use dehydration synthesis. So dehydration synthesis requires an enzyme, and it produces removes a molecule of water to join two monomers together. We saw the same thing when we joined our fatty acid chains onto our glycerol molecule to make triglycerides. So the removal of a molecule of water to make covalent bonds using an enzyme, so dehydration. And we saw it also in the last session where we joined two nucleotides together through dehydration synthesis, removal of a molecule of water to make what we called a phosphodiester link. It's no different with proteins. So what we can see up here is one amino acid. So here's the alpha carbon and its hydrogen. Here's the R group. Here's the amino group. Here's our carboxyl group, our carboxylic acid group. And notice here's our hydroxyl group that is part of that carboxyl group. And then over here, we've got a second amino acid. So alpha carbon and hydrogen, R group, carboxyl group, amino group. And here we can see a hydrogen, one of those hydrogens attached to the nitrogen. And what do we get when we have H2O? So an enzyme will come along. It will break off the hydroxyl group of this carboxyl group. It will break off that hydrogen from the amino group. H2O gives us a molecule of water that is given off. And then a new covalent bond is formed between this carbon and this nitrogen, which you can see here with this blue line. And that joins our two amino acids together. And this link or bond is called a peptide bond. So a peptide bond is a bond that forms between amino acids, and it's formed through dehydration synthesis. When you eat your lunch later, or you eat your dinner later, and you have protein in your food, remember from our first session when we started talking about this, those proteins are going to be far too big for us to use. They first need to get broken down into their monomers, into their amino acids. So how do we do that? We'll carry out the process of hydrolysis. So again, we use enzymes, and an enzyme will add a molecule of water. It will break this peptide bond, and it will add an hydroxyl group back here, the hydrogen back here, and it will separate back to our two amino acids. So we build our molecule, we build our proteins through dehydration synthesis, and we break them down through hydrolysis. So this is just the process that we use to join amino acids together, and this is what's happening at those organelles called the ribosomes. So the ribosomes are reading the instructions that's in our messenger RNA that we talked briefly about last week. It is then sending out for the amino acids to be brought into the ribosome via the transfer RNA, and then the ribosome acts as the enzyme. The ribosome is basically joining these two amino acids together through the removal of a molecule of water. So hopefully by this point you should start to be comfortable with dehydration synthesis and hydrolysis. We have talked about it a few times now. If not, so sort of just go back over your notes, and if in any doubt, just ask. So what do we get then? Well, if we join two amino acids together like we just did, so we can see that example up here, two amino acids, remove a molecule of water, and we join through a peptide bond. Well, that's called a dipeptide, di meaning two. Two amino acids combined as a dipeptide. If we were to add another amino acid, well, we would call that a tripeptide, tri for three. Once we start getting into the realm of four or more, well, we're then moving into the polypeptide. So a polypeptide is basically a long chain of amino acids. So we can see here. Notice that, like I mentioned earlier on, this is the backbone. Okay, so this is the backbone of our amino acids. So if we look at this, here's the alpha carbon, here's our amino group, here's what's left of our carboxyl group, here's the side chain, here's the peptide link joining it to this amino group, the alpha carbon with its hydrogen, the carboxyl group, peptide link, amino group, alpha carbon, and so on and so forth. So one thing that I didn't mention, I'll quickly go back to this previous slide, is that amino acids will only ever join together through the carboxyl group of one amino acid and the amino group of the other. And that comes down to electrical charges. So the carboxyl group of an amino acid will carry a slight negative charge. And again, this goes back to something that we've discussed before, and it's called electronegativity. So because, remember I said that oxygen is very, very electronegative. So when you get these shared bonds, these covalent bonds between the oxygen and the carbon, well, the shared electron sits slightly closer to the oxygen. So the oxygen here carries a slight negative charge. When we start looking at the amino groups of amino acids, the nitrogen is also quite electronegative. It's quite a highly electronegative atom. So the shared electrons between the nitrogen and the hydrogen, the electrons sit slightly closer to the nitrogen. That leaves the protons in the nuclei of these hydrogens slightly exposed. So the hydrogens are slightly positively charged. So the amino groups are slightly positive, or these hydrogens are slightly positive, and the oxygens of the carboxyl groups are slightly negative. So that is why these two are attracted to each other, the negative to the positive. So you will never find two amino acids joined together, amino group to amino group, or carboxyl group to carboxyl group. It's always the minus and the plus coming together. Join our amino acids together through dehydration synthesis, and for each peptide bond that is formed, a molecule of water is given off. So here we can see a diagram that just shows us the process of protein synthesis. And I briefly went through this with you last week, but if we look at this diagram, we can see our section of messenger RNA, so this long strand here. This was copied from our DNA, so copied from a gene, which is a part of a chromosome that carries the instructions for building a protein. So our messenger RNA will combine with the ribosome, so that's this blue unit here. And the ribosome will go along and it will read the codons. So if you remember, codons are 3 adjacent nucleotides that code for the placement of an amino acid in a protein. So the ribosome will read the codons on the messenger RNA, and then we can see these sort of ribbony things here are our transfer RNA. The transfer RNAs will bring the amino acids into the ribosome. They match up, so you can see that they also have codons on there. These are actually called anticodons because they match up with the codons on the messenger RNA. And they deliver the amino acids to the ribosome, and then the ribosome will facilitate dehydration synthesis, joining the amino acids together to make those peptide bonds. And what we can see here is the formation of a polypeptide, so this long chain of amino acids. So that's how we go from the instructions in the DNA to our polypeptide, our long chain of amino acids being formed. So messenger RNA carries the instructions that it copied from our gene in our DNA. The ribosome reads the instructions in the messenger RNA. Transfer RNAs bring the amino acids into the ribosome based on the instructions within the messenger RNA. And then the ribosome acts as an enzyme to carry out dehydration synthesis to join our amino acids together via dehydration synthesis to form those peptide bonds. This is a process that is called translation. So the process of copying the instruction from DNA to make this strand of messenger RNA is a process called transcription. And then this process of turning those instructions into a long chain of amino acids is called translation. So ribosomes translate the instructions in messenger RNA to create a polypeptide, so a long chain of amino acids. and it's made of these amino acids in a specific sequence. That bit is incredibly important. So these amino acids are not put together in any old order. It is very specific based on the instructions that are written in the order of the codons within our messenger RNA. So what we ultimately end up with at the end of this is a polypeptide. So that's a long chain of amino acids. But there's an important distinction to make here, and that is this is not a protein yet. So a polypeptide is not a protein. It's just a long chain of amino acids. It will not become a protein until it has undergone a highly complex process of folding and twisting and coiling. And it is that process of folding that creates the unique structure that will enable that protein to carry out its specific function. Translation produces this long chain of amino acids, a polypeptide, but then that polypeptide will undergo a pretty intense sequence of twisting and folding and coiling to make a very complex three-dimensional structure that will be then, we can class it as the protein. It will have the right shape to do its job. And if we go back to this same diagram that I showed you right at the beginning, this is a protein. So you can see here, this is much more than just a long chain of amino acids. There are lots of repeating patterns in here. We have all of these areas of spirals. We have these areas which are denoted by these arrows that are sort of parallel sections where the polypeptide is folded back over itself. So this is a protein. It's got a complex three-dimensional shape. And when we look at this complex three-dimensional shape of a protein, what we can see is that they are complex molecules, and they typically have 3 or 4 different levels of structure that appear within them. So when we take a complex three-dimensional protein such as this, when they're studied, we tend to find repeating patterns within them. And we actually call these the levels of protein structure. And the 4 different levels of protein structure that we can see down here are primary, secondary, tertiary, and quaternary. Now, all proteins will have a primary, secondary, and tertiary level of structure, but some proteins are complete at this section, but then some proteins will go on then and have this 4th level of structure, this quaternary level. So what we need to look at next is, well, I'm going to go through each one of these levels of structure with you, and I'll just talk to you a little bit about what it is. All proteins start off with this joining together of amino acids then that we've just looked at. We refer to the unique sequence of amino acids that make up a protein as its primary level of structure. It's really, really important because it is what ultimately will determine how the protein folds up into its three-dimensional shape that will ultimately give it its three-dimensional structure that enables it to do its job. And as we talked about earlier, there are 20 different amino acids, and these 20 amino acids can be joined together in any order. And that order is what is determined by our DNA. That is what the sequence of nucleotides in our DNA does. It codes for this unique sequence of amino acids. But I refer to it as, or think of it as, letters of the alphabet and how they come together to make up words. So we've got 26 letters in our alphabet, and you can join those letters together in an almost infinite amount of ways to make up hundreds of thousands of words that make up the English language. Well, it's the same way with amino acids and proteins. We've got 20 different amino acids, and you can join them together in specific orders to make specific proteins. If you don't join them together in exactly the right way, you won't get a protein that is folded into the right three-dimensional shape and that will mean it won't be able to do its job properly. The primary level of structure then is this unique sequence of amino acids, and that will ultimately determine how the protein will coil and twist and fold into its three-dimensional shape. So I'm just going to show you this animation now to go back over primary structure.
Each protein has a unique primary structure, a particular number and sequence of amino acids making up the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. This illustration shows some of the amino acids making up the primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone, that makes each of the 20 kinds of amino acids unique. Different amino acids have different properties that affect the folding of a protein. Thus, primary structure ultimately determines the shape of a protein, which determines its function.
So that's the primary level of structure then. The next level of structure that we see within proteins is called the secondary level of structure, or secondary structure. There's a couple of important things to know about this. The first thing is that secondary structure is defined by patterns of hydrogen bonds between backbone amino and carboxyl groups. So what does that mean? Well, if we go back to the initial or the original structure of a generic amino acid, if you can picture that in your mind, we had an alpha carbon in the middle, and then we had an amino group to the left, which, as I said, was positively charged, and then we've got the carboxyl group to the right, which is negatively charged. Now, if you can imagine, you've got a long chain of amino acids that, let's say it's 1,000 amino acids long. As the polypeptide chain starts to fold back over itself and coil around itself, you will start to get hydrogen bonds, so attractions between the positively charged amino groups and the negatively charged carboxyl groups. And what we find is that when we get repeating patterns of these, we start to see two specific shapes emerge. And those two specific shapes you can see on the diagram here. When we get sections of parallel lines forming, so this is where the polypeptide will start to fold back over itself, we get these hydrogen bonds that you can see here. So this is a hydrogen from the amino group, so this would be positively charged. And then we've got this negatively charged oxygen from the carboxyl group of this amino acid. And these dotted lines are the hydrogen bonds, so that will help to fix this in place. And we can see more here and more here and so on and so forth. These parallel sections are what are referred to as beta-pleated sheets. We also get a second type of structure that appears within secondary structure, and that is these areas of spiral or coils that tend to form. And again, they're just held together by hydrogen bonds between those backbone elements. So here's the positively charged hydrogen of the amino group, because you can see the nitrogen there. Here's the oxygen of this carboxyl group here. So this is negatively charged, and it's holding together through this hydrogen bond. And when we get these types of spiral or coils, we refer to them as an alpha helix, or plural alpha helices. So when we start to look at the secondary level of structure within a protein, we tend to see these sections of alpha helix and beta pleated sheet, and they are held together by hydrogen bonds between those backbone elements. So let's have a quick look at secondary structure.
In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself or where two regions of the chain lie parallel to one another. Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, giving them partial negative charges and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself.
Okay, so now we've got primary structure and we have secondary structure. That will take us on to tertiary structure. So tertiary structure is really important because it's where we start to get our overall three-dimensional shape of our protein. So now we're moving from the realms of just a polypeptide chain, so a long chain of amino acids, to now getting into the realms of a protein with a complex three-dimensional shape that is now able to do a specific job. And the key thing about the tertiary structure, besides the fact that it's this three-dimensional shape, is that it's caused by interactions between the R groups or the side chains of all of our different amino acids. If we remember, secondary structure is caused by hydrogen bonds between the backbone elements of the polypeptide. Tertiary structure is interactions between those 20 different R groups that we talked about earlier. And because those R groups are so varied, they're so diverse in their molecular structure, we get quite a few different types of bond and interaction that can form between them. So I've put some examples here. You don't need to worry too much about them. It's useful to remember some of them, but it's more important that you realize or you remember about tertiary structure, that it's the overall three-dimensional shape of the protein, and it's created by interactions between the R groups. So although it's useful to remember some of these, I would focus more on the basics that I've just mentioned, those two main points. But just to give you some examples then, we can get hydrophobic and hydrophilic interactions. So sections of amino acids that are hydrophobic, well, they will turn away from water and they may bond together to get away from water. Hydrophilic sections or hydrophilic amino acids will tend to move towards water so they can bond with water. We get these weak interactions called van der Waals interactions. We also get other weak bonds, so hydrogen bonds and ionic bonds. And then we do get some stronger covalent bonds like disulfide bridges that form between sulfur atoms that are in certain R groups. So we get a pretty diverse range of interactions between the R groups of the amino acids. And that's what basically helps to form this complex three-dimensional shape. So let's have a look at the animation for tertiary structure then.
Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall three-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups.
Okay, so that's tertiary structure. And like I said just now, some proteins are complete at this level. But there are some proteins, particularly larger ones, that will have a fourth level of structure, and that's called quaternary structure. So quaternary structure is quite a nice, easy one to remember, actually, because it is just a result of when two or more individual polypeptide chains join together to form one protein. Another way of looking at it is if you could imagine two or more proteins with a tertiary level of structure, and then they join together. And it's that joining together of multiple proteins that will give us our quaternary level of structure. And it's just held together by the same types of bonds between R groups that we see in tertiary structure. So a couple of good examples, and one that we can actually see here in this diagram. So one is hemoglobin, which we can see here, and another is collagen. So these are examples of proteins with quaternary levels of structure. And if we look at hemoglobin, what we can see is that it's actually made up of 4 individual proteins, or what we would actually call polypeptide subunits. But we don't need to worry too much about that. We can see one up here, one over here, and then one here, and one down here. So we've got 4 separate units that make up this protein to give it its quaternary level of structure. So let's have a quick look at the animation for quaternary structure.
Some proteins consist of 2 or more polypeptide chains. The 4th level of protein structure, quaternary structure, results from the combination of 2 or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, the red oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of 2 kinds of polypeptide chains. 2 of each, a total of 4 chains, make up each hemoglobin molecule.
There we can see our 4 levels of protein structure then. So hopefully that shows you how we go from simple amino acids, and then we join our amino acids together through dehydration synthesis to make peptide bonds. That will ultimately give us a long chain of amino acids that we call a polypeptide. That polypeptide will then go through a process of folding and twisting and coiling to give us a complex 3-dimensional shape or structure. And it is that complex structure that enables the protein to serve its function or to do its specific job. So it only takes 1 or 2 of the amino acids in the primary level of structure to be wrong, and that can lead to all kinds of complications. So as an example, you might be familiar with a disease called sickle cell anemia. And sickle cell anemia is a misshapen protein within the structure of hemoglobin. And it is caused by just 1 amino acid being wrong. So 1 amino acid is incorrect in the primary structure of that makes up hemoglobin, and it gives you red blood cells that are misshapen. So it doesn't take very much for a protein to be no longer able to do its specific job. And that brings us on to something called denaturing or denaturation of proteins. So because these proteins are so complex in terms of their structure, and they are held together by such a variety of different bonds and interactions, it means that they are quite susceptible to damage. It means that they're quite fragile molecules. Our bodies, and this is true of all life, goes through an awful lot of effort to try and maintain an environment that is suitable for the healthy functioning of our proteins. And this is something that we'll explore in later lessons, and we'll actually talk a little bit more about in the next session when we look at enzymes, because as we know, most enzymes are proteins. But certain things that will affect the structure of our proteins are temperature, so particularly high temperatures. So high temperatures will cause the bonds and interactions that stabilize secondary, tertiary, and quaternary levels. It will cause those bonds and interactions to undo. So our protein will essentially unravel. And that unraveling of the protein structure is what we call denaturing or denaturation. Other things that will cause denaturation of proteins are things like pH. So hopefully you're familiar with that term. So pH is the measure of acidity or how basic a solution is. Our proteins need to be within a specific pH range to maintain their structure. If a solution becomes too acidic or too basic, it will cause the bonds and interactions to undo, causing the protein to denature or unravel. And then it will lose its structure and it won't be able to do its job. So keep that in mind. This is something that we're going to talk a lot more about in next week's session, where we look at enzymes. Sometimes if the exposure to the incorrect temperature or pH isn't too dramatic and it's a temporary change, as long as the protein can quickly go back into its original environment, it can actually go through a process of renaturation. So it will actually fold back up into its original shape. So that's the process of denaturation and renaturation. But if you expose proteins to excessive heat, so a good example of this would be if you fry an egg or if you boil an egg. So if you were to crack an egg into a cold frying pan, it's liquid. The egg white, as you now know, is called ovalbumin, or contains a protein called ovalbumin, and it's white. It's transparent. And that is because those proteins in their proper form are soluble in water. But what happens when you start heating that frying pan up is that you cause the proteins to denature. And as they denature, they unravel. And in that unraveled state, they are no longer soluble in water. So they precipitate out of solution and solidify. And that's what we see as a fried egg. When you've got that solid white structure, that is that is caused by the denatured proteins. And this can make certain proteins easier to digest. And that's one of the reasons that cooked food is really useful. It makes it easier for us to, or our bodies, to access the nutrition within it, as well as sort of killing off pathogens and things like that. So that's denaturation. Keep that in mind. That's going to become really important in the next session when we look at enzymes. I didn't want to spend too much time on this, but it's worth mentioning because we've just talked about this process of coiling and folding and twisting to create this complex three-dimensional shape. So how does that happen? And this process of protein folding is pretty complex, and just by looking at the primary level of structure, so the unique sequence of amino acids, it's very difficult for scientists to predict how a protein is going to fold up and what its ultimate structure is going to be. And this is a pretty important area of scientific research at the moment. It's trying to look at the genetic structure, or the nucleotide sequence within DNA, and then trying to work out what protein, in terms of its structure and then ultimately its function, we're going to get from that particular gene. So it's a pretty complex process though. And that's because most proteins will go through several stages on their way to becoming a stable structure. So some proteins will automatically fold up as they're produced at the ribosome, but others will need to be moved into certain parts of the cell where the conditions are right for them to actually establish their full structure. And we actually have specialized proteins themselves within the cell that enable them or assist in this proper folding. And you can see them here. They're called chaperonins, and they're quite interesting. And they're one of those things that we see inside cells that are a bit of a head scratcher, because you think, how did that ever evolve? But what you can see here, so this is a chaperonin, and it's basically like a little capsule. So it's got a little cap on top, and then it's got a hollow cylinder. And what will happen is the cap will come off. Our polypeptide that's been produced at the ribosome, so this long chain of amino acids, will go inside. The cap is then replaced, and then inside the chaperonin now, we have an environment that is conducive to that protein being folded up into the right structure that it needs to do its job. And then once that folding is completed, the cap comes off, and then you end up with this properly folded protein emerges, and then it can go off and do its job. And what we do know is that certain really important diseases are caused as a result of improperly folded proteins. So I talked about the impact that having incorrect amino acids will cause. So we talked about sickle cell anemia, but we can also get things like Alzheimer's and Parkinson's disease, and also mad cow disease. these are often associated with misfolded proteins. So remember what I said right at the beginning, structure equals function. If the protein isn't the right structure, it won't be able to do its job properly. So if you don't fold that protein correctly, it won't be able to do its job. That was a bit of a whistle-stop tour of proteins. And like I said, we're going to recap on that or talk about them in a bit more detail next week when we look at enzymes, because enzymes are a particularly important part of protein, a particularly important type of proteins. So let's just do a quick recap then on them, see what you remember. So I'll read through the questions, and then we'll pause this, give you a chance to write down your answers, and then we'll go through the answers together. So number 1, how many essential amino acids are there? You can give yourself an extra point if you put down how many amino acids there are in total, and then how many essential there are. Number 2, what makes each amino acid different? Number 3, how do 2 amino acids join together? So what is the process? And then what is eliminated? And then number 4, name the 2 forms that we find in secondary structure of proteins. And then number 5, what is a protein's quaternary structure? So have a go at answering those. I'll pause this now, and then we'll come back and we'll go through the answers together. Okay, welcome back. Hopefully you've had a chance to answer those questions. So let's go through the answers together now. So question number 1, how many essential amino acids are there? Well, there are 9. So there are 9 essential amino acids, and hopefully you also remembered that there are 20 amino acids in total, so 20 naturally occurring amino acids that make up our proteins. 9 of them are essential, which means that we cannot synthesize them in our bodies, we have to get them in our diet. Number 2, what makes each amino acid different? Well, it's the R group or the side chain. So the backbone of the amino acid is always going to be the same, that alpha carbon with its hydrogen, and the amino group and the carboxyl group, that's always the same, but it's what attaches to the alpha carbon, the R group or side chain. Number 3, how do 2 amino acids join together, and what is eliminated? Well, hopefully you would all have got this now, because this is the 4th time that we've talked about it. It's dehydration synthesis. And dehydration synthesis forms our peptide bond between amino acids, and for each peptide bond that is created, a molecule of water is given off or is eliminated. Okay, number 4, name the 2 forms of secondary structure. Well, they are alpha helix or alpha helices and beta pleated sheets. And remember, secondary structure is created by hydrogen bonds between the backbone elements of the polypeptide. And then number 5, what is a protein's quaternary structure? Well, it's when 2 or more polypeptides join together to make a single protein. So hopefully you did okay with those. If you didn't, or if there was some there that you got wrong, then don't worry at this stage. Just go back over your notes. You can watch this again, or have a look at the textbook. If this is your first time looking at this, it's quite a lot to take in, and that's absolutely fine. So these quizzes are quite useful, not just for checking what you know, but also for checking what you don't know, so you know which bits you need to go back over. So that brings us to the end of our session on proteins then. So we started off by having a quick recap of the functional groups. Then we looked at amino acids, the 20 different amino acids with their backbone structure and their R groups. We started joining amino acids together to form peptide bonds. And then we looked at protein structure, so how we got from single amino acids all the way up to quaternary levels of structure. And then we also talked a little bit about protein folding and also denaturation. So hopefully that was useful. Like I said, we're going to recap on some of this again in the next session when we look at enzymes. Okay, so hopefully that was useful. Thanks very much. I will see you in the next session.